Hello everyone, welcome back. So this is the week 5 of the NPTEL course, Experimental Biochemistry. And this course is coordinated by Professor Das Gupta from the Department of Chemistry and Professor De from the Department of Bioscience and Biotechnology. And I am Bias Mukherjee, a Prime Minister's Research Fellow from the Department of Bioscience and Biotechnology. And today we will discuss about chromatography. Okay, in this class. Now, chromatography is a very important technique for all your different kinds of exams, be it GATE, be it CSIR NET, be it DBT, be it ICMR. So, all of these different exams plus the NPTEL examination. So, we will discuss about a lot of different chromatographic techniques. We'll talk about affinity chromatography, size exclusion chromatography, ion exchange chromatography, HPLC, hydrophobic chromatography, and a lot of different chromatographic techniques. And we'll try to solve MCQ questions, MSQs, and a lot of numericals. Okay. So if you have any doubts, you can always pause me in the middle and you can ask questions. You can answer the questions in the chat box. Hopefully, we are going to have a good, valuable class. Okay, let's start. Right. So we'll start talking about biomolecules. Now, what are biomolecules? Biomolecules are biological molecules that perform a lot of different functions in our body. So in our body, all of these major biomolecules are actually divided into 4 different types. Okay. So one of them makes up the fat portion in our body. So they are the lipids. Next comes the nucleic acids. Okay. So nucleic acids are DNA, RNA, and different types of DNA and RNA molecules. So those make up the nucleic acid portion in our body. The next one is carbohydrate. Now, carbohydrate, we all know these are sugars, right? So they are sugar molecules, different combinations of different kinds of sugars. These are carbohydrates. And then we have proteins. Now, we have been talking about proteins in the last 2-3 classes. So you guys are already familiar with proteins by now. Okay, so amongst all of these, only one type of biomolecule is actually less than 1000 Dalton or 1 kDa. Okay, so these are the lipids. They are biomicromolecules. All the others are called biomacromolecules or macrobiomolecules because they are larger in size. They are all greater than 1 kDa. Okay, now there's something called bioseparation. What is that? Bioseparation is the technique that is used for separating out all of these different biomolecules from a mixture. Okay, so when we want to study the biomolecules individually, we will have to devise techniques to separate them out because this is very essential for any kind of uh, further in-depth study of these biomolecules, or if you want to make something new out from them. Okay, so you want to engineer them or you want to use them specifically for one particular study. So for that, separation is one of the most essential techniques. Okay, so we will start with bioseparation. Now, what is bioseparation? Bioseparation refers to the process of isolation and purification of biomolecules from complex mixtures such as cells, tissues, and fermentation broths. Okay, the goal is to obtain highly pure and active substances. This is very important. They should be functionally active, and that is why choosing the specific kind of bioseparation technique is very essential. So we need to choose something that will first of all help us to get the pure form of a particular biomolecule plus active form. Okay, and use for various applications such as biotechnology, pharmaceuticals, and research. Okay, now here is a table for you. So different kinds of biomolecules are separated based on different properties. Okay, so for example, for lipid separation is majorly based on solubility and based on polarity or hydrophobicity. So what are the techniques? There are different techniques. You can read about them. So there is chloroform methanol separation. So you can read about this. Since our course is concentrating more on the other biomolecules, the biomacromolecules, so we will talk about those in today's class. So we'll majorly talk about chromatographic techniques. Okay, but you can study about this separation of lipids as well if you're interested. Okay, then we have thin layer chromatography. So we are going to talk about this in today's class. And there are other specialized methods, for example, ultracentrifugation. So ultracentrifugation means spinning at very high speed and separating them out, okay, based on the size. Now we have sugars. So what about sugars? So precipitation of sugars can be done using ethanol precipitation method. Then gel filtration chromatography can be performed for separating out the sugar molecules based on their size. Okay. Then we have paper chromatography, thin layer chromatography, gas chromatography, HPLC. All of these are based on the polarity or hydrophobicity of the sugar molecules. Then we have based on charge, we have electrophoresis. Okay, so this can also be performed for sugars. And we have affinity chromatography technique. So there's lectin. Lectin is carbohydrate binding proteins. They are called lectins. So these lectins can be used for isolation of specific carbohydrate molecules. And then there is anion exchange chromatography, which depends upon the charge of the different sugar molecules. Okay, now we have nucleic acid. So from here, we will be talking about this in details in the next class. Okay, so phenol chloroform extraction method is commonly used for DNA. Trizol method. So trizol is phenol chloroform plus guanidine isothiocyanate. So that is GITC. That method, so it is called trizol because there are three different layers that get formed in the separation. So we will talk about all of these in details. Okay, so these are the places from which you can expect a lot of questions because these are very, very crucial and these are very basic techniques that all of you must remember. Okay, so trizol extraction is one of them. The next one is separation based on molecular weight. Now you already possibly know this. That is agarose gel electrophoresis, which is done for nucleic acids. Okay, most very commonly done for nucleic acids. Next we have based on charge, there is ion exchange chromatography. Then based on specific affinity. So for example, let's say that this particular, let's say particular mRNA, it has a poly A tail, right? So what is poly A? Poly A means many adenine are there, around 250 adenines are there at the tail of the naturally occurring mRNA, right? So then we can have these complementary probes which are having poly T. So it is called oligo DT. So deoxythymidine. So it is a stretch of these deoxythymidines and these will have affinity for this poly A tail and that is why mRNA will be stuck to the chromatographic column. Okay, so that is poly A tail targeting oligo DT chromatography, which is a kind of affinity chromatography. Right. So next we have cesium fluoride density gradient centrifugation. So we will be talking about all of these. Okay. Now we have for proteins. Now this we will be talking about in today's class. Right. So there are different methods. There is ammonium sulfate precipitation, which is salting out. Now we will talk about salting in and salting out. I think we have talked about salting in, salting out in the previous class. If not, then we will be talking about this in either today's class or the upcoming class. Okay, but yeah, we are going to talk about all of these in details in this course. Okay, so then we will talk about gel filtration chromatography, which is also known as size exclusion chromatography. And then there is SDS page, which is separation based on size. Both of these are separation based on size. Then we have ion exchange chromatography, native page, and we have isoelectric focusing. So all of these depend upon majorly the pi of the protein. Okay, and native page also takes into consideration the size of the protein, the pi of the protein, the molecular interactions of the protein, because here the protein is run in the gel without denaturing it. So its native properties are all retained and the separation is done based on the native properties. Okay, now we have hydrophobic interaction chromatography. So this takes into account the one sorry. Okay, so this takes into account the polarity of the molecule. So we'll talk about this in details again. Right, and we have affinity chromatography, which can use his-tagged proteins. Now you know that recombinant proteins are made. Recombinant proteins are given specifically the 6 histidines at the N terminal or C terminal. That is called the his tag. Now this his tag has affinity for nickel or cobalt in the chromatographic column, and that is why his tagged proteins are purified using affinity chromatography using Ni-NTA columns, that is nickel bound to nitro. There is this NTA compound, we'll talk about that. Okay, so that is how this is done. Then we have 2D page. So 2D page is two-dimensional page. So in one dimension, the separation is happening based on the pi, and the other dimension, the separation is happening based on the size. So we'll talk about this also either in today's class or in the coming class. Okay. So let's continue. Hopefully that was not too much for you people, and hopefully everything did not go over your head. So let's start. Now we have all of these at a glance. We'll just look at them once again and brush them up. Okay. So separation principles based on solubility, lipids, proteins and nucleic acids are separated based on solubility. Then charge. So proteins and nucleic acids are separated based on charge, right? Then there is size. So proteins and DNA, RNA, they are often separated based on size. Polarity, lipids and sugars are often separated based on polarity. Specific binding, so mainly proteins and mRNA, they are normally separated based on specific binding. Okay. And then again the chart is given. So the different kinds of techniques that are used for different kinds of biomolecules for their solubility, size, charge, polarity, affinity and density based separation. Okay, so you can go through all of these. Now we have discussed this in the previous slide, so I'm not repeating this, and it is just for you to remember things. Okay, so now we will start with chromatography. Okay, so let's try to understand. Chromatography is a very important technique, and chromatography is a very interesting technique. Okay, so all you need to do is just try to understand it, and you'll be able to solve questions based on this. Okay, so it's very simple, very interesting, a little complicated at places, but it's okay, we can manage. Okay, so what is chroma? And okay, so the word chromatography comes from chroma, meaning color. Okay, chroma, that is color, and graphene, which means to write. So chromatography is a biophysical technique used for the separation of mixtures into individual components based on their differential properties. Okay, the term chromatography is derived from the Greek words chroma, meaning color, and graphene, meaning to write. Now if you look at these two figures, okay, so you will see that there is a strip of paper which is having a mixture of different colors, and you dip it into a solvent, and as the solvent is soaked up by this paper, the solvent rises and the colors are also rising and they are getting separated out. So that is what is happening in this paper chromatography, right? So chromatography is employed in the purification of biomolecules such as proteins, nucleic acids, for use in biotechnology and medical research. It finds applications in pharmaceutical industry for quality control, ensuring the purity of drug formulations. So in environmental science, it aids in monitoring and analyzing pollutants in air, water, soil samples. Moreover, chromatography is essential in food and beverage testing, forensics, and the study of complex biological processes. Right, so chromatography has applications in diverse disciplines. So that tells us that this is very, very important as a technique. Okay, now we will talk about the common terms in chromatography. One second. Yeah, so we'll talk about the common terms in chromatography. Okay, so firstly, there is analyte. What is analyte? All the things that you want to separate. Okay, so analyte is the substance that is to be separated from the mixture during chromatography, right? Now we have the mobile phase. So what is the mobile phase? Mobile phase is the component that moves with the sample. Now the sample is there in the mobile phase, it moves. It is either a gas or a liquid that passes through a column where the components of the mixture are adsorbed or absorbed. Okay. Then there is the stationary phase. So what is the stationary phase? Inside the column, whatever is there to absorb the mobile phase, that is called the stationary phase, or that is called the okay. Yeah, so these lights have sensors. You give me a second, I'll be back. Yes, good. So we have the light back again. Now, stationary phase is the one that doesn't move with the sample. It is generally a porous solid that absorbs the components from the mobile phase. Okay. Then we have eluent. Now, what is eluent? Eluent refers to the fluid that enters and passes through the chromatographic column. Okay. And what is elute? Elute is the fluid that contains the sample. So elute means that it is now eluent means everything that is put into the column, right? It contains the analyte. And what is the elute? So elute is the final protein, the final desired product containing solution that comes. That is my elute. Elute is the fluid containing the sample that exits the chromatographic column. What is elution? Elution is the process of removal or extraction of a solid by washing it out with a suitable solvent in a chromatographic column. It does not necessarily need to be a solid, it can be a liquid also. Okay. Now, resolution. So what is resolution? Resolution means, let's say I have two fingers here, right? So how well can you see that these two fingers are two distinct fingers? So it is basically how well can you distinguish between two dots that are very closely spaced. That is called resolution. So the degree of separation between two adjacent peaks in a chromatogram, that is called resolution. So more the resolution, better the purity, and more extensive purification has happened, more expensive that instrument, right? So that is all about resolution. Now we have peak. So what is peak? Peak means that a graphical representation of a separated component in a chromatogram. So that means the intensity is absorbance is actually recorded of a particular solution that initially starts coming and then a lot of it comes out and then it again very little is coming out at the end, right? So this is one peak. That means one particular type of thing has come out of the column. Now we have the principle of chromatography. So what happens in chromatography? Okay, so the separation technique of chromatography is dependent on the principle of partitioning the constituent compounds into two phases. One is the mobile phase, the other one is the stationary phase. So whatever goes inside the column, this either this is the mobile phase, now this binds to the stationary phase, right? Now the sample, your protein of interest, let's say, this can either have more affinity for the stationary phase or it can have more affinity for the mobile phase. If it is having more affinity for the mobile phase, it will flow through and it is called flow through. Now if it has more affinity for the stationary phase, then it will go and bind to the stationary phase and then it will be eluted later when you give some very high salt buffer. Okay, so analytes having higher absorption. So see the method of, okay, one second. The mobile phase is used to indicate the mixture of the substrate that is to be separated and dissolved in a liquid or gas. The stationary phase is used to describe a porous solid matrix through which the sample present in the mobile phase percolates. Right. So the method of chromatography is based on the fact that the dissolved substances in the same solvent may have different solubility, right? So that is the major principle. And the solute with the greatest solubility rises faster and segregates from the mixture. So that means it is not binding to the stationary phase, it is getting flown away first, right? So the different solubility of the analyte is due to different affinity or different strength of adhesion towards the mobile and stationary phases. Now the affinity of the analyte can be explained in two terms. First one is adsorption and the second one is solubility. So adsorption is the ability to get attached to the surface of the stationary phase, while solubility is the ability to dissolve in the mobile phase. Now analyte having higher adsorption will slowly pass to the column. It will very slowly pass to the column. Analyte having high solubility will move faster through the column. So the difference in solubility and adsorption is due to the difference in polarity between the different solvents, between the different phases, which determines the extent of adsorption and solubility. For example, a polar analyte will adsorb better to a polar stationary phase. So let's say the stationary phase is positively charged and the analyte is negatively charged. It will obviously get stuck, right? But if the stationary phase is positive and the analyte is also positive, it is definitely going to flow through. Okay, so that is the principle of anion exchange chromatography. Okay, so it can depend upon the polarity, it can depend upon the charge, it can depend upon a lot of different things. Affinity for the specific like stationary phase, etc. Right, so for example, a polar analyte will adsorb better to a polar stationary phase and thus moving slowly through the column, and a non-polar analyte will better dissolve in a non-polar mobile phase, thus moving faster in the column. So that is the basic principle of the stationary phase and the mobile phase and the partitioning between these two phases. Okay, hopefully you have understood. Now we'll move on. So until now, whatever we have talked about, we will see that here. Okay. So the components of the analyte interact differently with the stationary phase and the mobile phase based on their nature, right? Based on their polarity, based on their charge, based on their property of attraction, adhesion, solubility, etc. Right. So depending upon their polarity, they spend more or less time interacting with the stationary phase and are thus retarded to a greater or lesser extent. This leads to the separation of the different components present in the sample. So each sample component elutes from the stationary phase at a specific time called as the retention time. So when it is coming out, that is its retention time, and as the components pass through the detector, their signal is recorded and plotted in the form of a chromatogram. Okay, so here the example that is shown for you, this is for paper chromatography. Okay, so what is happening? You can see that from the origin, all of these different colors are rising to different heights, and it is like different. So the distance traveled by the sample divided by the distance traveled by the solvent. So you can see the solvent front. That means how much the solvent has been absorbed. So the ratio between the two is known as the retention factor. Okay, now in very basic numericals, they'll ask you to find out the retention factors, but in other numericals, they'll ask you to find the partition coefficient. So what is partition coefficient? We'll look at it. Okay, right. So now we have partition coefficient and the relationship between this retention factor and partition coefficient. Okay, so what is partition coefficient? Partition coefficient is the concentration of the analyte that got differentiated, distributed between the stationary and the mobile phase, right? So concentration of the solute in the stationary phase divided by concentration of the solute in the mobile phase. That is called the partition coefficient. It is referred to as K or KAV, right? So what is K? Larger K means more solute prefers stationary phase, that is slower movement. So if it is stuck to the stationary phase, that means it is moving slowly. But if it is more soluble, then it is going to move very high up with the solvent, right, in the mobile phase. So now relationship between RF and partition coefficient, you can see that RF is equal to 1 by 1 plus K beta, where K and beta, they are both given, right? And RF is equal to, distance traveled by the solute divided by distance traveled by the solvent front. So RF value changes from 0 to 1. Larger RF means that moves more and prefers the mobile phase, so it is more soluble. Smaller RF means that it is preferring the stationary phase and it is sticking more to the stationary phase. So this is something that you can keep in mind. Okay. Right. So now we will talk about the different kinds of chromatography techniques. So chromatography can be classified into different types based on the following categories. Yeah. So based on, one second. Okay, so chromatography can be divided into different types based on the following categories of physical state of the mobile and stationary phases. Okay, so chromatography based on the bed, Now what is bed? Bed is what is there inside the column. Okay, so based on the bed, there can be column chromatography, there can be planar chromatography. So there is no column, right? There is only a sheet of paper. So the examples are paper chromatography and thin layer chromatography. These do not have a column, these just have a sheet of paper, right? And in thin layer, you can see what is this thin sheet made up of. Next. Acha. So based on the physical state of phases, there is gas chromatography and there is liquid chromatography. So for liquid chromatography, the two different types of liquid chromatography that we can talk about are high performance liquid chromatography, which is HPLC, and then there are also other different types of liquid chromatography. Right, so next we have, based on the mechanism of separation, there can be affinity chromatography, which is Ni-NTA, right? One of the examples is Ni-NTA based chromatography. Then there is this oligo-DT chromatography, using which you separate this mRNA. And then there is ion exchange chromatography. So these can be again of two different types. One is ion exchange chromatography and the other one is anion exchange and cation exchange chromatography. The next type is gel filtration chromatography, which is the third type. Okay, so gel filtration is also called size exclusion chromatography. Now these are the different techniques given to you for viewing at a glance. So see, in case of gel filtration, what happens is, based on the different sizes, the molecules get separated out. What about hydrophobic interaction chromatography? So we will talk about all of these in details. Okay, so anion exchange is based on charge-charge interaction. Hydrophobic interaction is again based on polarity and hydrophobicity. Affinity chromatography, you understood, Ni-NTA is one type. The other one is whether this particular substance has special affinity for this other substance or not. Then based on that, this is done. Then there is reverse phase. So what is reverse phase? Using the property of polarity, we are ensuring that one particular analyte binds to the stationary phase at one condition and it detaches from this particular stationary phase at a different condition of the buffer. So we'll talk about all of these in details. Okay, so we have the different types of chromatography techniques at a glance. Okay, right, then we will start with the MCQs and then we will look at the chromatography techniques in details after this. So we have a table here and then after that we look at them in details, but before that we will solve a few questions. Okay, so what about affinity chromatography? So see, there is a specific biological interaction that is exploited here. For example, ligand and target binding. So there are different kinds of these examples, right? Right. So there can be glutathione and glutathione S transferase interaction. There can be lectin and carbohydrate interaction, right? So lectin is a carbohydrate binding protein. So all these different kinds of interactions that can be there. Then we can have oligo-DT and mRNA interaction. So the stationary phase is often the ligand immobilized on the bead. For example, Ni-NTA, and then there can be some kind of antibody with which our protein interacts, and the protein here is the antigen, right? So then the mobile phase is the buffer, specific buffer, and best suited for highly specific proteins, his tag proteins, antibodies, enzymes, etc. Elution method is of two different types. One is biospecific elution, otherwise high salt elution. So what is that biospecific elution or competitive elution? So what are we doing? So we will talk about this in details. So basically you know about this his tag, right? So histidine, in the structure of histidine, it has the side chain. So the side chain is called imidazole ring. Now what we are doing is we are adding a lot of free imidazole rings, okay, free imidazole rings in the buffer, and it is huge in number, and then we are flowing that buffer through. Then what happens, since these imidazole rings, they are more mobile, they are smaller in size, they are very highly mobile, so they will go and they will compete with these binding sites, they will compete with the histag proteins for binding to this nickel. So they will push off the histag proteins and they will bind to nickel, and our histag protein is going to elute. This is one type of elution, competitive elution, biospecific elution. The other type of elution is increasing the salt concentration. So when the salt concentration is high, then what happens? This interaction is reduced because, okay, one second. This is the issue with teaching in the classroom. Yeah, right. So we will continue. Now, what happens is these are the different kinds of elution. So here what happens, the interaction between the nickel and the imidazole ring, these are reduced because of the increase in salt. Okay, so that is something that happens. Now, this is useful for protein purification, mRNA isolation, etc. Next, we have size exclusion chromatography. Now, let's say that your protein, now you have performed Ni-NTA based purification, you have the protein, but unfortunately your protein is having some other protein bound to it. Okay, so now what do you do? So now what you can do is, let's say that you are, so besides your, okay, no, not bound to it, let's say that your protein is there, but there are some fragments, some broken fragments of that same protein as well. So those also got bound to his tag, right? So some N-terminal fragments or C-terminal fragments containing the histag is there. Now, I want the pure protein. I don't want these fragments. So, what do? I will go for size exclusion chromatography. Now, the fragments are definitely of different size. So, based on the resolution, we will be able to separate the fragments out. How do we do that? We have this column and the column has porous beads. Now inside the beads, inside the pores of the beads, the smaller fragments will enter and my protein will enter into the larger pores of the bead. So what happens is my protein is going to elute faster compared to the smaller fragments because they enter into more number of pores and they take a longer time to get eluted. So always remember in size exclusion chromatography, your protein of interest, if it is very large, it will elute first. If it is of medium size, it will elute in the middle. And if it is very small, it will elute at the last. So it's always in this order. Okay. Now, what happens is molecular size and hydrodynamic volume. So what is hydrodynamic volume? Let's say that this is a protein, okay, and surrounding the protein the water molecules are arranged. Now the radius is not just from this center to this end, it is not that, but the radius is from this center to this center of the water molecule which is surrounding it. That is called the hydrodynamic radius, and this entire volume is called the hydrodynamic volume. Okay, so based on the size and hydrodynamic volume, we are separating them. Then the porous beads. So beads are made up of superdex. Okay, what is superdex and what are agarose? You can read about that. These are made from natural substances. There are sephardex, there are superdex, all of these different. Oh God, this is very irritating, but unfortunately we will have to deal with it. Give me a second. Okay, so we'll continue. Now we have understood about this size exclusion chromatography. Then there is protein saccharide complexes. So these are all useful for separation using size exclusion chromatography. And then the elution method is isocratic. So there is no gradient. So gradient means that there is a mixing of two buffers in which one buffer is initially 0% and the other buffer is 100%, and then this gradually increases and this gradually decreases. But that is not happening here. It is just a single buffer. Okay, there is no gradient elution required. And then It is very useful for changing the buffer of a protein. So it is in one particular buffer, you want to change it into another buffer. So it is very useful for that. Then checking whether there are any aggregations, whether there are any fragments, separation of those fragments, or separation of two or more different conformations of the protein. Let's say one of the protein conformations, like there's one type of fold and then there's another type of fold of the same protein, then we'll be able to, if the size is different of the two foldings, then we will be able to separate them out. Okay, now we have ion exchange chromatography. Here what happens, the net charge of the molecule is taken into account, and the charged resins like DEAE, which is diethyl aminoethyl, which is charged, and then there is CM cellulose, which is carboxymethyl cellulose. Okay, this is negatively charged. DEAE is positively charged cellulose. So these are used as resins. Buffer with increasing salt gradient is used, and proteins and nucleic acids are separated out using ion exchange chromatography, and salt gradient or pH gradient buffers are used for elution. Okay, salt gradient means that initially it is 0 salt and then there is 1 molar salt, and these are mixed such that this gradually increases and goes to this 1 molar salt. Okay, protein purification and isoform separation is done using this. Then we have hydrophobic interaction chromatography. So this takes into consideration the surface hydrophobicity in high salt and mildly hydrophobic ligands, phenyl, octyl, etc. These are useful to be separated. So these are all stationary phase. Then high salt buffer is used, and decreasing salt is employed, and proteins are useful for separation using this method. And then decreasing salt gradient, 1 second. Okay, you give me 5 minutes. We'll be back after 5 minutes. Okay, and then we'll continue with this. So until then, you can look at this table and you can read about this. I'll be back in 5 minutes. Hello everyone, welcome back. So here we will not have this issue with lights, hopefully. Right, so let's continue. So we were talking about the different kinds of chromatographic methods. And the last one that we have is reverse phase HPLC. So this is again utilizing the polar nature of the solvents. Okay, so what is normal phase, what is reverse phase? We will talk about all of this in details. Right, so we will start with a few questions, but before that we have a few more methods that we need to look into. Okay, so reverse and normal phase HPLC, let us look at this. Right, so what happens in reverse phase HPLC? Let's see. So the hydrophobicity is the strongest interaction here, and non-polar silica, for example C18 and C8, these are used as stationary phase. Water plus organic solvent is used for mobile phase. Acetonitrile or methanol is generally used. Okay, now we will have peptides, small proteins and lipids, all of these separated using HPLC, reverse phase HPLC, and this is useful for peptide purification and analytical separation. Right, now we have normal phase HPLC. Now how is it different? Here also we take into account the polarity of the molecules. Here we have polar silica, mind that. There we had non-polar silica, here we have polar silica. Okay, and non-polar solvents are separated using normal phase HPLC. Okay, then we have lipids and small organic molecules, polar solvent polarity change, etc. All of these are elution methods, and lipid separation is done using normal phase chromatography, HPLC. Now we have HPLC technique, which is high pressure liquid chromatography, and it depends on C18, ion exchange, etc. So you can go through all of these. Now there is thin layer chromatography, there is paper chromatography, all of these different techniques are there. So right now we will start with the questions and answers. Okay, so for a small brush up, we have this tiny chart for you. So hydrophobic interaction chromatography versus reverse phase HPLC. So in hydrophobic interaction chromatography, we have mild hydrophobic interaction, the proteins remain folded, but in reverse phase HPLC, very strong hydrophobic interaction is there and it often denatures the proteins. And ACC versus SDS page, both use mass and size based separation, but in ACC the protein is in its native form, but in SDS page the protein is getting denatured. Okay, now we have ion exchange versus isoelectric focusing. So in ion exchange we have separation by charge at a given pH, and in isoelectric focusing the separation is based on the pi of the protein. Okay, so separation is based on the pi of the protein. Right, so now we will start with the question and answers. Okay, let's start. So question on separation techniques, you can answer in the chat box. Okay, so a particular protein can be separated from other contaminating proteins by which of the following methods? So we have mass spectrometry, we have fractional distillation, we have okay, C. Great. Let's see. Let's look at the right answer. Yes, fantastic. It is C. Okay, the right answer is C. Now we have another question, which is if the partition coefficient in double, then by what factor is the molecular weight changing? Okay, so for a particular protein, if the partition coefficient is seen to have doubled, then how has the molecular weight actually been affected? A, let's see. Okay, so the calculation is done here, you can see. So the partition coefficient in chromatography represents the distribution of a solute between two immiscible phases, the stationary phase and the mobile phase. It is also known as the distribution coefficient or retention factor. Okay, Now K is equal to concentration of solute in stationary phase divided by concentration of the solute in the mobile phase. Now if K becomes 2 times, then which are the components that are not changing? So A and B both are constants and it is e to the power minus mw. Right, so then that is becoming, how is mw getting affected? So then you get log inverse of this K, and then the final answer is ln into molecular weight. Right, so if KAV is double, then how is it changing? It is basically changing in this particular way. Okay, because it is square. Okay, now we will try to understand size exclusion chromatography. Now this is a very nice diagram explaining everything to you, right? So there are these big particles which are eluting first because they are not getting into the pores of the beads. Then there are these medium sized particles which are only getting into some pores of the beads, so they are eluting later. Then the tiny particles that are getting into all the pores of the beads, and that is why they are eluting at last, right? So one of the type of partition chromatography is gel filtration chromatography or size exclusion chromatography. And in gel filtration chromatography is based on the principle that molecules are partitioned between the stationary phase and mobile phase that consist of a porous matrix. Now the molecules with larger size than the pores, they do not enter into the porous beads and elute the fastest. They make up the void volume. So what is the void volume? Void volume is the volume of the liquid outside the beads. Okay, so outside the bead, all the white area that you can see in the figure, that is the void volume. The medium sized molecules elute next as they enter only some pores. The smallest traverse through the pores for the longest duration and they elute the last. So partition coefficient or KAV is equal to VE, that is volume elution minus V naught. V naught is the void volume divided by VT, that is volume total, minus V naught is once again the void volume. So this formula you should remember. Okay, it's very important. So in the next slide, this is explained in more details. So you can see that KAV is equal to VE minus V naught by VT minus V naught, where VT is the total volume, VE is the volume at which the molecule of interest elutes, assuming that it is within the fractionation range of the matrix. V naught is the volume of the space between the beads or the void volume, and Vi is the volume of the space within the beads. Vi is not taken into this equation, right? So V total is the volume outside the beads plus inside the beads. Okay. Now the applications of gel filtration chromatography are molecular weight determination, fractionation of the molecule, separation of fractions, desalting, that means changing the buffer, equilibrium, binding, buffer exchange and dialysis. Okay, right. Now we will try to understand something even more interesting, and that is called affinity chromatography. So here what we are seeing is one antigen and one antibody, they are interacting. They interact by very specific interactions. For example, there can be hydrogen bond interactions, there can be ionic bond interactions, hydrophobic interaction, van der Waals interactions, etc. So in nature, there are a list of things that interact with these very special interactions with their counterparts. These interactions are taken into consideration for this particular type of chromatography technique design. So affinity chromatography is a biophysical technique that exploits the unique biological function. Give me 1 second. Yes. So now we don't have this extremely annoying sound. Okay. So we don't have the noise anymore now, which is good. Let's carry on. So now, so what happens is all these unique biological interactions are taken into consideration for designing this particular technique. So the chemical structure of the biomolecule such that it specifically binds to an immobilized ligand reversibly and can be separated from a mixture of several biomolecules. So the biological interactions exploited here are listed below. So antigen-antibody interaction, enzyme-substrate analog, not enzyme substrate because then the substrate would be used up, right? So we need something that doesn't get used up. So analog, binding protein and ligand. Then we have receptor ligand interaction. Then we have lectin and polysaccharide or glycoprotein, right? Then we have nucleic acid and complementary base sequence. We have hormones and vitamins and receptor carrier proteins. Glutathione and glutathione S-transferase, which is an enzyme. Metal ions, nickel and cobalt. And we have polyhis fusion proteins or native proteins with histidine or cysteine on their surface. So even native proteins can have uh, a stretch of histidines and they can bind to these his tagged, right? So nickel, that is one issue with, it's a drawback of this particular technique. Okay, now we'll look at NINTA based purification. So this pink thing that you're seeing, this is the protein. Now protein is having these imidazole groups of histidine projecting out. These form this coordination complex with nickel or cobalt. Nickel or cobalt is bound to the agarose bead using NTA, which is nitriloacetic acid. Right. So NINTA based affinity chromatography to purify proteins recombinantly produced using a hexahistidine tag or naturally rich in histidine amino acids, also called as immobilized metal affinity chromatography or IMAC. So since a metal has been immobilized here, so this entire technique is called IMAC or immobilized metal affinity chromatography. Right. Now nickel is covalently coupled to agarose using NTA. NTA is a organic thing which is hexadentate ligand that coordinates Ni2 plus and histidine residues in the analyte protein that is naturally or recombinantly synthesized with the His6 tag bind to this coordination complex. The binding is attributed to the intrinsic property of histidine to bind to nickel and cobalt. FPLC, which is fast protein liquid chromatography, that is the full form of FPLC, which is an important question in your exams. This exploits this NINTA affinity chromatography technique to purify the his tagged proteins. Okay, so elution is either biospecific means with analyte like competitor. So I told you about imidazole, right? And or non-specific, for example, increasing pH or salt concentration. Okay, the next technique that we have is understanding ion exchange chromatography. So the figures are such that you will understand everything from the figure. So if you look at this, you will see that in cation exchanger, the resin is what? Negatively charged. So the molecules that you are having, they are cations. That is why they are positively charged. So the resin is CM cellulose and the molecules are coming and binding. And then what happens is anions and neutral molecules, they are flowing by at first. Then we are using uh, ions which are positively charged and they are coming and binding to the resin and the protein is getting displaced and it is eluting. So ion exchange chromatography is based on the reversible electrostatic interaction between charged biomolecules or analytes and oppositely charged functional groups on the stationary phase matrix, that is ion exchange resin. Elution is carried out by increased concentration of the counter ions of the same charge as the analyte. So in cation exchange chromatography, CM or carboxymethyl cellulose, the charge of the resin is negative. What type of buffer is used? Acetate buffer, citrate buffer, phosphate buffer, which can have very what pH? Low pH of around 4 to 7. What about anion exchange chromatography? Anion exchange chromatography uses DEAE, which is diethyl aminoethyl cellulose, which is positively charged. And the buffer that is used has a very high pH, for example, tris buffer, acetate buffer, phosphate buffer. Okay, they are having pH of 7 to 10. Okay, great. Now, there is something called hydrophobic interaction chromatography. So you can see that the hydrophobic surfaces, so there is this highly ordered water shells that surround the hydrophobic surfaces of ligands and proteins, and the hydrophobic surfaces are forced to merge and minimize the total area of such shells to maximize the entropy. Salts enhance the hydrophobic interaction, and the equilibrium of this hydrophobic interaction is controlled predominantly by salt concentration. So you know about salting in and salting out. So what is salting in? When you add little bit of salt and it is actually helping the interaction, that is called salting in. So it is helping in more solubilization, but salting out means that it is helping in more hydrophobic interactions, and uh, salt interacts with the water molecule so that the protein and the ligand can interact with hydrophobic interactions. So we will look at this in details now. So see what is happening. So the hydrophobic surfaces are there in both the protein and the ligand, and it is surrounded by ordered water shells. Now water forms a structured cage-like arrangement around the hydrophobic regions. This ordering decreases entropy. So this water is now having lower entropy because it is not having randomized movement. Correct. So now the system wants to increase the entropy. Why? Because delta G is equal to delta H minus T delta S. So if the entropy is higher, then the delta G term will become lower. Then what will happen? It will be a spontaneous and very stable system. Now when the hydrophobic patches come together, then what happens is the water molecules that were there, they get displaced. So entropy of water increases. As a result, the overall term becomes, overall delta G becomes more negative, and so this is the interaction is getting favored. Right, so the total hydrophobic surface exposed to water decreases. The ordered water molecules are released, water becomes less ordered, entropy increases, and entropy gain drives hydrophobic interaction. So this is what we had studied even in our protein folding class. Correct. So hydrophobic interaction is entropy driven, not bond driven. Enthalpy is due to making and breaking of bonds, but entropy is because of displacement. Okay, so it is driving hydrophobic interactions. Right. Now we will see what happens when there is salt in the picture. So when salt concentration increases, what happens? Salt ions compete with water molecules. Water becomes more engaged in solvating salt ions. So now salt is interacting with water. Then what is happening? Less water is available to solvate the hydrophobic surfaces, and that is why hydrophobic patches become more exposed. So the proteins bind more strongly to the hydrophobic ligands. This is called salting out effect. So it is helping in protein-protein interactions rather than protein-water interaction. And high salt means binding increases, low salt means binding decreases. And high salt means strong hydrophobic interactions, protein binds to column. Low salt means that weak hydrophobic interactions and proteins elute. Okay. Now we will talk about HPLC. So what is happening in HPLC? See, the separation in the reverse phase HPLC is based on hydrophobic interaction between the non-polar analytes in relatively polar mobile phase and non-polar stationary phase. So the stationary phase and analytes, they are going to interact. The mobile phase is going to flow through. So analytes will be higher hydrophobicity. Analytes with higher hydrophobicity will interact more strongly with the stationary phase and will take longer to elute from the column. The desired analyte is eluted by gradient elution, where the composition of the mobile phase is changed during the analysis. Application is separation of nucleic acids and other biomolecules with very high affinity. So we will look at this in more details now. Okay, so you can see here that the blue circles, they are the pillars or the stationary phase. Yellow is the flow of the mobile phase, that is solvent under very high pressure. Separation depends upon different affinities of the components for stationary and mobile phase. Okay. Now affinity for the mobile phase is stronger for that we can see for component A. So what happens is component A prefers the mobile phase. So it is more soluble in the mobile phase. It spends more time dissolved in the solvent and it elutes first. It is not binding to the stationary phase, so it is eluting first. It travels faster with the solvent stream. It has shorter retention time. So it is my flow through. Now if the component B is very much non-polar, then what is happening? Component B is binding to the stationary phase and it is interacting more with the stationary phase, so it is flowing very slowly. It will have a greater retention time. It will elute later, right? So in normal phase HPLC, the stationary phase is polar, typically silica, and the mobile phase is non-polar. So the polar molecules remain stuck, the non-polar molecules elute. For example, lipids. So separation occurs based on polarity, that is polar compounds interact more strongly with the polar stationary phase, so they are retained longer and elute later, while the non-polar molecules elute first. In contrast, reverse phase HPLC has a non-polar stationary phase, commonly C13 or C18, C8 and C18 bonded silica, and the polar mobile phase, the water mixed with methanol or acetonitrile. This is my mobile phase. Acetonitrile is very harmful for our body. That is why HPLC is a very expensive instrumentation, because it requires that high pressure, plus it is also very difficult to uh, use because of this very toxic acetonitrile and methanol used as mobile phases, right? So whereas the polar compound elutes first and the non-polar elutes later. So the key difference lie in the polarity of the stationary phase and the mobile phase and the order of elution. So in normal phase, more polar elutes later. In reverse phase, more polar elutes first. So we will discuss the questions. Okay, now, so start answering the questions. This is a graph given to you. Which of the following statements is or are true for the above graph for gel filtration chromatography? A and B are more distinctly separated than C. B is a dimerized form of A. D. Okay. Molecular weight of C is lower compared to, yeah, very. Yes, that is correct. D is absolutely correct. So since C is eluting much later than A and B, that means C is having much less molecular weight compared to A and B. So small molecules can enter the entire intra-particular pore space and hence elute later compared to the larger molecules, which are excluded from all pores. Therefore, they elute earlier. Okay. Now, void volume in a gel chromatography column refers to, what is void volume? Space within the matrix pore, the volume of the column which is unoccupied by matrix granules, volume of the protein added to the column. C. Very good. Correct. Yes. So void volume V naught is the space outside the matrix granules. Very good. Okay, now we have a small question. So it's a numerical question. In size exclusion chromatography, Sephardex G200, this is the column name, and what does G200 mean? That which molecular weight, up to what molecular weight it can separate, and all of these. Okay, so G200 is useful for separating a specific range of molecular weights of proteins. So you can look at this. Okay, you can read about this. So there is S75, S600, S200, S300, all of these different types of columns. So the column volume is 60 ml and volume of the stationary phase is 45 ml. If the elution volume of the solute is 60 ml, then find the distribution coefficient Kd. Okay, so you're saying that it is 0.63. Very good. Let's see what is the right answer. Achha, so the right answer here is 0.44. Why is that 0.44? Because C. We know the formula is KV, that is partition coefficient, is equal to VE minus V naught divided by VT minus V naught, right? So V naught is given as the void volume, which is 1/3 of the column volume. Always remember, V naught is often 1/3 of the column volume, which is 20 ml. VE is given as 40, and VS. So what is VS? VS is the volume of the stationary phase. So that is 45 ml. So KD is the distribution coefficient, and it is VE minus V naught divided by VS. Right? So VS is basically VT minus V naught. That is only VS, and that is coming to be 0.44. Okay. I hope that is clear. Okay. Now we have another question. Okay. So if there are any doubts, you can ask me. Achha. Right. So question on chromatography. 3 different proteins are to be separated for analysis. The characteristic of the proteins are given below. Protein A, B and C. They have 3 different molecular weights and they have pi. Okay. Now which of the following would result in the best separation of these proteins to get them in the pure form? We can do paper chromatography, we can do gel filtration chromatography, we can do SDS page followed by ion exchange chromatography, and we can do ion exchange chromatography followed by gel filtration chromatography. So which technique do you think would be best for the separation? D. Acha. Acha. So no, it's not going to be D. It is going to be gel filtration chromatography only. Why? Because if you look at the PI, all of that PI are very close by. Right, so if the PI is that close by, then using any buffer, can you make sure that one of the protein is having positive charge and the 2 other proteins are having negative charge? It is very difficult to do that. Right, so whatever buffer condition you keep, all the 3 will be having same charge. Then you cannot separate them using ion exchange chromatography. So that is why, since the molecular weights are very different, it is best to separate them using their size exclusion chromatography. Okay, and why not SDS page? Because for SDS page, you cannot get the protein in hand. You can just see what different proteins are there. So SDS page will actually, it will not give you the protein in hand, right? So separation using SDS page is not going to work here. So that is the thing. So see, ion exchange cannot be used since their pi is very close. Since these proteins are largely differentiated based on their molecular weight, therefore the best technique for separating them would be gel filtration chromatography. Okay. Now we have another question. Carboxymethyl matrix and DEAE matrix are generally considered as cation exchange matrix, cation exchange and anion exchange respectively, anion exchange matrix, anion exchange and cation exchange respectively. So how will you do this? This is very simple to remember. Carboxy means negative charge. For anion exchange, what should be the charge of the matrix? For cation exchange, what should be the charge of the matrix? Diethyl amino ethyl. Amino means positive charge. B. Okay, very good. Let's see. Yes, correct. So B is the right answer. Right. So for carboxymethyl, since it is COO minus, it will be positively charged cation exchange matrix. Correct. Because only then the cation will bind to it if it is having negative charge. Okay, we'll go to the next question. So before loading the impure protein solution, it is in phosphate buffer pH 7.4 in the Ni-NTA column. What will you do? So in the next class, we will talk about protein purification probably. So then you'll see that we will discuss all of these different steps in protein purification. Okay, so right now what you can do is, which of these will you do before loading an impure protein solution? So remember that the columns that we have, they are very sensitive. So one of the things that you can do, should do in fact, is definitely centrifuge, filter and then only then you have to load it. Okay, so centrifuge the solution at 11,000 rpm and discard the supernatant. Centrifuge the solution at 11,000 rpm, discard the precipitate. Check the pH using a pH meter. Equilibrate the column with the mobile phase, that is phosphate buffer 7.4. So what do you think needs to be done? So a few of these need to be done. So which all do you think need to be done? Okay. So this is the basic practice for every FPLC system. Okay. Now, so we look at the answer, no problem. So the right answer is B. Why is that B? Because the protein, okay, so the bacterial protein that you're growing, you are sonicating so that the bacteria is broken open and the protein is coming into the solution. And you spin it. Why do you spin it? So that all the bacterial debris, so the broken pieces of the bacteria and the nucleic acid and all of these cell debris, all of that goes and settles down as the pellet. In the supernatant, there is the soluble portion. So the protein can be there, the nucleic acid can be there, but then you have used DNAs, so the nucleic acid has been chopped off. Okay, so all the flow through is basically all the supernatant is what is the soluble part. So what you do is you take the supernatant, you throw the pellet away, and you take the supernatant and then you check the pH of the supernatant. That is not very necessary, we don't do it in the lab, but here since it's there in the question, so I think the answer should be 0.63 because the correct answer is 0.56 and it is closest to 0.60. Correct me if I'm wrong. Okay, sure. So I'll look at this question then. So which question is this? Let's go back and check once. Okay, and then I can confirm and I can tell you. Acha. Like, okay. So it is this question, right? Okay. So I will check and then we can discuss this in the next class. Right? You can even mail me once just for a reminder in case I forget. Okay. So and then we can, go through with it and we can check. Okay. All right. Yeah. So we were talking about this one. So let's continue talking about that and I'm definitely going to check that. Don't worry. Okay. Right. So what we do is we check the pH if we want to and we filter it. So then what are the different steps? You see that you have to discard the precipitate, that is going to happen, and you have to check the pH and you have to equilibrate the column. So whenever you are running a protein through a column, you need to equilibrate the column with the buffer that the protein is in. So yes, option number B would be the right answer. Okay. Now we have another question. Which of the following statements is or are true? So in membrane-based filtration method, commonly used to remove higher molecular weight compounds for a common anion exchange chromatography, sodium acetate buffer of pH 4.5 is used. In affinity chromatography, only one of the proteins bind to the column material based on the specificity and high affinity. In gel filtration chromatography, it can filter proteins based on size. So which of these do you think are correct? D. Fantastic. Let's see. Yes, that is very correct. So D is the right answer because the membrane-based filtration method is commonly used to remove lower molecular weight compounds for a common cation exchange chromatography. Sodium acetate buffer of pH 4.5 is used. The rest two are correct. Okay. Okay. We have a few more questions. We have a lot of questions today. Okay. So match the following. Try matching. Affinity chromatography, gel filtration chromatography, cation exchange chromatography, hydrophobic interaction chromatography. And we have the different matrix materials. There is silica gel, there is CM cellulose, there is Ni-NTA matrix and biogel P300, and we have DEAE Cephalose matrix. So how are you going to match C? Let's see. Yeah, very good. So C is the right answer. Perfect. Then we have another question. A sample contains a mixture of proteins from which protein X is to be isolated. Which method would be the accurate isolation method? So see, molecular weights are given and pi is given. So for which of the proteins are you going to do separation based on isoelectric point, and which proteins are you going to do separation based on gel filtration chromatography? So which of these options would be the right option? Ion exchange chromatography only. Ion exchange followed by gel filtration chromatography. Now X is, see, you can see that three of them have molecular weights very close by. Correct. And for ion exchange chromatography, what would be the pH? Okay, so if the pH is very different, then you can separate out three of them, and then you can. So this is how you can do it. And then affinity chromatography followed by ion exchange chromatography. Then we have gel filtration chromatography only. So which of these would be the best method to separate? B. Okay, let's see. Yeah, so B is the right answer because the pi are so different. That is why ion exchange chromatography can be done. And since X is having a pi of around 4.3, so then if you take it this way, that then what will happen is in the mixture there will be 4.3 and this 3. So basically A and X will come together, and then what you can do is, since A is having molecular weight much different from X, you can go for size exclusion chromatography. Yeah, so very good. Now you have your protein of interest contains 6X histidine tag and is overexpressed in bacterial culture. After purifying by Ni-NTA affinity chromatography, you run the eluted fractions by reducing SDS page to confirm its presence and purity. However, you observe, in addition to your protein of interest, an additional band. Which of the following is possible explanation for this? Right, so his tag got cleaved. The protein does not contain a tag. In addition to your protein, some bacterial protein may also contain tandem repeats of histidine, histidine residues, and the protein is multimeric. So after Ni-NTA, you are seeing bands in SDS page. What would be the conclusion? C. Yes, C is one of the answers. Right. So this hexahistidine tag is there. Out of those 6 histidines, 2 bind to nickel or cobalt, and there can be these natural proteins also that can have histidine tags and they can bind. Now we will try to answer questions on HPLC. Okay, so there is a mixture of these 3 compounds. One is benzene, one is phenol, one is benzoic acid. The separation is carried out using C13 reverse phase HPLC with water acetonitrile mobile phase. Now, which is the correct order of elution? Now, see, this is basically C13 reverse phase. So, in reverse phase, what is happening? The most mobile, the most polar one is going to elute first, and the most non-polar one is going to elute last. So based on your understanding from there, which one would be the right answer? Which is more polar and which is least polar amongst benzene, phenol and benzoic acid? A. Okay, good. Let's see. Yes, very good. A is the right answer. Why? Because you see the order of polarity is benzoic acid is most polar. Then it is phenol and then it is benzene. So definitely benzene is least polar. So it is going to elute at last. Benzoic acid elutes the first. Okay, great. Now we have another question. Again, reverse phase HPLC. Increasing the percentage of acetonitrile in the mobile phase will increase the retention time of the non-polar compounds, decrease the retention time of the non-polar compounds, increase the polarity of the mobile phase, strengthen the hydrophobic interactions. What is going to happen? And the second question is in which is true. So in normal phase HPLC, non-polar compounds elute last. In reverse phase HPLC, polar compounds elute last. So you're saying B is the right option for the first one. Okay, good. Try answering the second one. RP reverse phase HPLC uses polar stationary phase. Normal phase HPLC uses non-polar stationary phase. A. Okay, good. Let's see. Yes, very good. So B and A, these are the right answers in the two questions. Very good. So using acetonitrile, what is happening? This makes the mobile phase less polar. So it reduces the hydrophobic interaction and the compounds elute faster and the retention time decreases. Okay. In normal phase HPLC, the stationary phase is polar. Polar compounds are retained for longer. Non-polar elutes first. Fantastic. We go to the next question. So resolution in HPLC improves when particle size increases, flow rate increases excessively, column length increases, and stationary phase is removed. C. Okay. Column length increases. Very good. So we will talk about this in details now. Okay. So resolution is proportional to root of column length. Remember. So smaller particles also help, but increasing particle size reduces the resolution. Okay. Now we have another two questions. Proteins with a protein with pi equals to 5 is loaded onto an anion exchange column at pH 8. What happens when the protein binds to the column? What happens? So pi is 5 and the pH is 8. So then what is the charge of the protein? And this is an anion exchange column. So that means the resin is positively charged. So then what will happen? Will the protein bind to the column? Will the protein flow through? Will the protein precipitate? Or no conclusion possible? Now remember number line. In the number line, the left hand side had negative and the right hand side had positive. Always remember that for protein charge, it is just ulta. Okay. So you have positive when the pH is okay. B and C, very good. Okay, very good. You're very fast. Good. So in hydrophobic interaction chromatography, the protein binding increases when salt concentration decreases, organic solvent increases, salt concentration increases, and pH decreases. Okay, very good. Let's see. Yeah, so we have B and C as the right answers. Yes, very good. B and C are the right answers. Excellent. So at pH is more than pi, we have the protein is negatively charged. So anion exchanger binds negative proteins, thus it binds. Now in hydrophobic interaction chromatography, the protein binding increases when there is high salt. It enhances hydrophobic interaction and promotes binding. Low salt results in elution. Very good. Excellent. Okay. Now we have, I said B, but it shows A. Is it? Does it show A? Oh, you said B. Oh, sorry. I did not check. Right. So yeah, it should be A because it binds. Okay. So I praised you too much. Anyway, it's okay, good. But you're trying, that is very important. Okay, we'll go to the next question. Which of the following statements is or are true in the FPLC system? The flow rate is maintained at 2 to 5 ml per minute. Increasing the flow rate beyond this can cause column breakage. Now, this is a very practical question. Okay. Now, a large air pocket can enhance the protein purity. Okay. At 280 nanometer, we monitored the UV absorbance, pH, and conductivity of the protein. Okay. After elution, SDS page must be performed to check purity. Which of these are true? Remember, air bubble is the biggest enemy of any chromatographic technique. Okay, so you should be very, very cautious when it comes to air bubbles, when it comes to liquid chromatography techniques. So now you try to answer. I gave you the hint. Okay, so we will look at the, yeah, D. Okay, so B is the right answer because 1 and D are, 1 is correct and 2 is wrong. So 1, 3 and 4, all of these are correct. Okay, so in FPLC, we monitored the UV absorbance at 280 nanometer. We check the pH and the conductivity of the protein. So I'm not sure if we can monitor the pH because we can monitor the pressure. So I think, yeah, no, then 1 and 4 would be the right answer. So you're correct. Yeah, we cannot check the pH in the FPLC, but we can check pressure. So I'll change this because, yeah, 1 and 4 would be the right answer. Okay, yeah. So 1 and 4 would be the right answer. Okay. So FPLC columns can withstand a certain pressure generated by the flow rate of that buffer. If the flow rate is increased due to high back pressure, the column will get damaged and large air pockets can damage the machine. So yeah, so when you're studying from this PPT, remember it is 1 and 4 because 3 we cannot monitor the pH on FPLC. Okay, so we come to the next question quickly. So which of the following will improve the efficiency of the separation process in liquid chromatography? Increase in the sample size, increase in the column diameter, reduction in the sample size, increase in the column diameter, increase in the sample size, reduction in the column diameter, reduction in both column diameter and sample size. So which one do you think would be best to increase the efficiency of the D? Very good. Yeah, that's correct. So it is reduction. It is D is the right answer. So why is D the right answer? That is explained here. So this is given in very elaborate way. Okay, so there is this equation which incorporates three different concepts. One is eddy diffusion, one is longitudinal diffusion, and the third one is mass transfer. So what happens is when you get a very sharp peak, that is a good uh, peak and there is good resolution. But when you get this peak broadening, so there is this very broad peak. So what are the things that can cause peak broadening? There are these three things that can cause peak broadening. One of them is called eddy diffusion, the other one is called longitudinal diffusion, and the third one is called mass transfer. So what are these? We will have a look at them. So see, in chromatography, band broadening and therefore loss in efficiency arises from three main processes described by the Van Deemter equation. So this equation that you see, H is equal to A plus B divided by mu plus C into mu, where H is the plate height. So what is plate height? This entire column is imagined to be divided into different tiny sections. Each tiny horizontal section is called a plate. So height of these each plates and mu is the linear flow velocity. Okay. So one of the term is called A term in this equation A, that is eddy diffusion, which results from multiple flow paths. So if the particles are very small, they can take multiple flow paths through the packaged system of the column. Different molecules take slightly different routes of unequal length and can cause peak spreading. So this term is independent of the flow rate and is minimized by uniform and small particle packing. Okay. The next one is longitudinal diffusion. So what is this? It is happening along the longitude. So due to natural molecular diffusion along the length of the column, this is the length of the column, so that is the longitudinal diffusion. So from high to low concentration within the band, it is significant at lower flow rates because the solute spends more time in the column, allowing greater diffusion, and thus its effect decreases as the flow rate increases, that is B by mu. So that is your longitudinal or lengthwise diffusion in the column. The next one is C, which is mass transfer. This arises because the molecule requires finite amount of time to equilibrate between the mobile and the stationary phase. At higher flow rate, the equilibrium cannot be established efficiently, causing the additional banding, right? Band broadening. This effect increases with the velocity c mu. So the interplay of these three factors produce this U-shaped band emitter curve with optimal flow rate at which the plate height is minimum and the column efficiency is maximum. So here all the equations are given for your better understanding. Okay. So what is this theoretical plates? Theoretical plates or chromatographic column is imagined to be divided into many small equilibrium zones called theoretical plates. More plates means better separation, narrow peak means higher efficiency, and it is given as H is the height of the plate. It is given as L is the length of the column divided by the number of the theoretical plates. Right, so smaller H means better column efficiency, larger H means poor efficiency, and high N and low H, and that gives you sharp peaks. And N is equal to 16 into T to the power 16 into TR divided by WB, where WB is the peak width at the base and TR is the retention time. So all these are different formulae that you must remember for solving the questions. Then we have something called peak broadening. Why does that happen? The three different components we already talked about are eddy diffusion, longitudinal diffusion, and mass transfer. So what happens is longitudinal diffusion decreases when the flow rate increases, but mass transfer increases when the flow rate increases. So there has to be this balance, and the particles should be smaller in size and they should have preferably similar size. Then the eddy diffusion would be reduced. Okay. So these are the different terms, and then there is the Van Deemter equation, and then the Van Deemter curve tells you that this U-shaped curve is formed, which is because of band broadening. At low flow rate, what happens is H is very large and longitudinal diffusion dominates. But at very high flow rate, mass transfer dominates and H is very high. But at intermediate flow rate, this is very, very optimal. H is minimum, and minimum H tells you that it is more maximum efficiency. Okay. So we'll go back to the question. We will see that reducing the column diameter in chromatography can improve the separation efficiency and resolution. The relationship between the column diameter and separation efficiency is given by this equation. You can see mu is equal to F divided by pi into T squared by 4, where F is the flow rate of the mobile phase. Okay. And we come to the last slide, which tells us that the term B by mu represents the contribution of longitudinal diffusion and C mu represents the contribution of resistance to mass transfer. Both of these factors are inversely proportional to the linear velocity mu. Therefore, reducing the column diameter D and decreases the linear velocity mu, which can lead to improved separation efficiency. Okay, that is all for today. We have one more question. You can try answering this, and this is the last question for today, and then we'll end the class. So a compound elutes 8 minutes, that is your retention time, in an HPLC column. The peak width, you saw that particular term, a peak width at the base is 0.4 minutes. The column length is 25 centimeter. What is the plate height? So you have to find out the number of plates and then you find out the plate height. Okay, how will you do? Very simple. So number of plates, you have the formula for number of, acha. So you're saying that it is A. Good. Let's see. Yeah, perfect. That is A, right? So N is equal to 16 into T to the TR divided by WB whole square. So TR is given as 8 and WB is given as 0.4, and the whole square you do, and then you get that as 6400, and then you calculate H from there, and so that is the right answer is 0.0039 centimeter. Okay, so that is all for today. If there are any doubts, we can discuss them in the next class. These are all the references. So here you can see that the virtual labs links are also there. So in this virtual lab training program, which I had shared with you previously in the last 2-3 classes, you know that there are all these pH, density gradient, etc. Proteins, peptides, SDS page, all these techniques are given. Similarly, experiment number 6 is, I think, affinity chromatography. Experiment number 7 is size exclusion chromatography. So you can go through those two, and there are all these questions that you can solve. Many MCQs are there. Can you suggest a topic for my poster presentation for Science Day in my college from chemistry or biomolecules? Very interesting. So, acha. So there are so many different things. So right now what is coming up is you can do something on AI and how AI is helping in designing these new therapeutics which are useful in, let's say, some kind of diseases, right? So one of them can be there if you're interested in AI, which can be a bit easy to explain. Acha. So you tell them like a story and it will be easy. Right. So if you understand it nicely, you'll be able to make them understand nicely. Okay. So there are so many different nice posters. So you can look at these. So if you ask me, since I work with proteins, I find proteins more interesting. So then you can like talk about, let's say you can talk about, there are so many nice topics. Poster presentation for Science Day in college. Yeah, you can actually just one. Acha, acha. Just one. Okay. Mm, yeah. So if you ask me, I would, since I work with engineering proteins and designing proteins, I would say you can like pick up one nice group of proteins of your choice that you are interested in. Let's say they are these DNA binding proteins or GPCRs. Now, GPCRs are coupled receptors, right? So these are used in around 50% to 70% of our therapeutics that we take, medicines, they are based on GPCRs. So you can talk about how these GPCRs are actually dominating the market, therapeutics market, and what kind of new research is incorporated into them. You can even talk about SARS-CoV-2 and like what are the different kinds of new strategies. So I'll give you one like interesting thing then. So basically in SARS-CoV-2 we all took this vaccines, right? But then a lot of people had a lot of complications in their body, and that is because these SARS-CoV-2 vaccines, they our body, our immune system saw them as like viral infection. So they saw them as a mimic of viral infection. So now people are designing a lot of different new types of strategies, new types of therapeutics and all that, where they can actually bypass all these problems that happened with the mRNA vaccines. So there are different kinds of new kind of vaccines that are getting developed. So these are all there. You can look up, you can suggest Gemini to give you good topics. Right. So yeah, something that interests you. It doesn't have to be very elaborate and very tough. It can just be like a small story that you like, you enjoy, and you are okay to tell others. Anything that you like. You can even talk. There is this very interesting topic for your Science Day. So right now you remember that people are actually bringing back all of these extinct animals to life. So you can make it like a debate that how they are doing this, how they are bringing these extinct animals to life, once again, and whether it is ethical or not, and whether it is important or not. Maybe there are some pros and some cons, right? So that is, it's based on molecular, like molecular biology, genetics, evolution, and then bioethics, and all of these different things come into play. I think Arctic fox or something was brought back to life, and now they are, I think, targeting a many other different uh, these animals and all. So you can see. So which one did you like? Yeah, it sounds interesting. Yes, and there are so many news articles also you will get how they are doing it, and so many people, they are actually revolting against this. So many people, they are saying that yeah, this is a very nice strategy, so we would actually get to experience real life Jurassic Park, the childhood movies and all, right? So that is a very interesting topic. Yeah, sure, thank you so much. I am glad that you like my ideas. You can like read about these, so these are very interesting. And then there are so many these new inventions. People are making water in these tiny balls which are consumable. So instead of carrying water bottles, so you can in fact actually talk about these products which people are doing. So there are many products, okay, so I'll tell you. So there is this water which is coming in small kind of a bubble type of thing. So you just take it, gulp it down, and you chew it and eat it up. It is not in a bottle, so it is not polluting the environment. Plus, there are also toothpaste is coming in the form of tablets. So these are all these new age discoveries, which you people can grow up and you people can become scientists and make a lot more of these kind of discoveries. Make sure to look up to the question. Yeah, sure. I'll definitely look up that particular question which you said, that particular MCQ. Yeah, okay, great. So that's it for today. Thank you so much for your attention. So if that's all, then we can end the class, and you can tell me how your science day went, National Science Day. Okay? Okay, okay, good, good, good. Bye bye. Okay, thank you so much. So we'll end the class today for now.